We chart it, we've described it, we've drawn it, we've mapped it. Now, just like the physical maps of our world that have been highly influenced by technology, think Google Maps, think GPS, the same thing is happening for brain mapping, true transformation. So let's take a look at the brain. Most people, when they first look at a fresh human brain, they say it doesn't look like what you're typically looking at when someone shows you a brain. Typically what you're looking at is a fixed brain, it's gray, and this outer layer, this is the vasculature, which is incredible around the human brain. This is the blood vessels. 20% of the oxygen coming from your lungs, 20% of the blood pumped from your heart is servicing this one organ. That's basically, if you hold two fists together, it's just slightly larger than the two fists. All of the sort of more complex thought, decision making is the last to mature, sort of a late adult. So if you take a deeper look at the brain, one of the things, if you look at it in cross-section, what you can see is that you can't really see a whole lot of structure there. But there's actually a lot of structure there. It's cells and it's wires all wired together. So about 100 years ago, some scientists invented a stain that would stain cells. And that's shown here. In the very light blue, you can see areas where neuronal cell bodies are being stained. And what you can see is it's very non-uniform. You see a lot more structure there. So the outer part of that brain is the neocortex. It's one sort of map brain function. We can start to tie these into the individual cells. So let's take a deeper look. Let's look at neurons. So as I mentioned, there are 86 billion neurons. There are also these smaller cells, as you'll see. These are support cells, astrocytes, glia. And the nerves themselves are the ones who are receiving input. They're storing it. They're processing it. Each neuron is connected via synapses to up to 10,000 other neurons in your brain. And each neuron itself is largely unique. The unique character of both individual neurons and neurons within a collection of the brain are driven by fundamental properties of their underlying biochemistry. These are proteins. They're proteins that are controlling things like ion channel movement. They're controlling who nervous system cells partner up with. And they're controlling basically everything that the nervous system has to do. So if we zoom in to even deeper level, all of those proteins are encoded by our genomes. We each have 23 pairs of chromosomes. We get one from mom, one from dad, and on these chromosomes are roughly 25,000 genes. They're encoded in the DNA, and the nature of a given cell driving its underlying biochemistry is dictated by which of these 25,000 genes are turned on and at what level they're turned on. And so our project is seeking to look at this readout, understanding which of these 25,000 genes is turned on.